Thank you. Thank you, Ronnie. And I again, on behalf of this team, I welcome you all to the second session, which deals particularly with some technical aspects of the seed storage in a community seed bank. So I really got some, I sent out a kind of a survey, or I would call it a pre-session, not an assignment, but a pre-session engagement. And many of you has already returned it back to me, and I could learn quite a lot that what exactly you are looking for, where you are currently, so that every one of us will be on the same page. So thank you for that. And we will also send, because some of you have asked some questions in that. Because today morning also I have received a few of your responses. So I'll just compile it to you and send it back to you. And based on those insights, I have modified today's discussion also a bit. So without further ado, I'll just get on with my discussion today. So what we are exactly going to do today is, let me see the, I get it. Okay. So what we are going to do today is to discuss about the seed storage basics. What are the basics of seed storage? What are the philosophy behind it, or in fact, the science behind it? And also the importance of seed moisture and how seeds are stored in a community, how seed should be stored in a community seed bank and how we do it. And we'll talk a little bit about the storage containers also, because today's session is much more about the technicalities. And adding to the technicalities, there are also documentations. So we'll be talking about the documentations also. So what are the enemies of safe seed storage? You might have seen my presentation earlier also, that primarily we say the temperature. Yeah, the temperature, higher the temperature, lower becomes the viability of the seeds over a period of time. Then of course the seed moisture, the humidity, that's one of the primary factors that affects the storage life of any kind of orthodox seeds. Orthodox seeds means not the taro, yam, or root crops, but the regular seed crops like the field crops or the vegetables or oil seed crops or pulses. So, and again, then comes oxygen. So higher the oxygen concentration, the storability also reduces. Of course, the initial vigor of the seeds, how the seeds initially, how vigorous they are the fresh seeds stored for a longer period of time. And of course, there are some physical aspects like the insects and the pests. And again, we will be discussing that how all these factors are really interrelated and how they affect the storability of the seeds. So talking about the seed storage basics. So how does these factors affect the seeds? So again, you can see the three primary factors: high moisture, high temperature, and high oxygen. So high seed moisture has actually two different kinds of effects. One is the physical and then the chemical. So on this orange box that you see here is the chemical aspect, that you know that high moisture water is a vehicle of all kind of chemical reactions inside the seeds. So more the water the seeds are getting, the faster they respire, respiration is higher, and faster they age. Faster they age, faster they die. It's just like any other living being. So high moisture causes faster aging. And also there are some physical things. There are mold infestation, because they find some water to grow on, insect infestation. Again, we'll be talking about insect infestation in a moment. And ultimately the seeds get damaged. So that's how seed moisture affects the storability of the seeds. And then comes the high temperature. The high temperature also acts in a similar way, that higher the temperature, within a certain range, of course, higher the temperature, the respiration rate is higher, higher enzyme activity, and again, the seeds age and die faster. And high oxygen, the pathway is a little bit complex, and it causes damage to the small cell organelles which are there within the cells. Within the seeds, there are cells, and these organelles are getting damaged due to the high oxygen. We won't go into much details about that, but in a nutshell, that high oxygen or high amount of air if you're providing to the seeds, it also causes the rapid death of the seeds and storability reduces. So that's how the whole thing is interrelated. So of course, but what we are going to talk about right now is the high seed moisture. We have just, I told you that how the high seed moisture is very much detrimental to the seeds. So now moving into let us see that exactly how important is seed moisture. So we have some, I'll give you a situation that we have a kind of a seeds which are onion seeds, which are having an initial germination content of 90%, and we are storing it under two conditions, A and B. So B is we are storing the seeds under in a cold storage, so 15 degrees, it's quite cold, but we have done only sun drying and the seed moisture content is around 10%. And the next situation is we couldn't get a cold storage, so we are storing it outside or inside a storage place where the temperature is not regulated, but in some way we could dry the seeds to 5% seed moisture content. So now let us do one thing. I'm going to you should be able to see a small poll on your screen. So what do you think? Under which situation the onion seeds will remain viable for a longer time? Is it the situation A or the situation B, or will both have the same results? So you will, I think I'm getting some responses. So please start clicking on that. You should be able to see a So under which situation the onion seeds will remain viable for a longer time? Will it be the one that is in the cold storage, that is B, or will it be the one which is in the outside or not in a cold storage, it is being stored under ambient conditions, but we found some way to dry the seeds a little bit. So what would it be? The cold storage or the hot storage? What do you think? Okay, so I can still see the things are coming up. Okay, I'll get back to your responses. I'm ending the poll right now and let us share the results. So this is the way you have responded. So let me correct what is going to happen. So this is what it's going to happen. So the seeds that were within the cold storage condition, their germination dropped from 90% to 40% within 2 years. But weren't these the cold storage conditions? But when we are storing the seeds under ambient conditions, but we are drying the seeds from 10% to 5% moisture content, the viability after 2 years drops only from 90 to 80%. So everything depends on this drying thing. Okay. So getting ahead, not just these chemical things that happens or the respiration and the aging within the seed physiology that goes wrong when the moisture content is high. Under high moisture conditions, the seed coats become soft. The seed coats are becoming soft, and it is the insects eat them easily, and their mouth parts are much easier to eat the seeds and damage the seeds for physical damage, under physical damage. So this is how also the moisture content of the seeds are affecting the ultimate seed quality. Now let us see what are the different kinds of seed storage, or what are the different kinds of seed storages that are available, and let us compare the costs, or let us compare the kind of investments that are necessary for these seed storage. So in this diagram, you can see that the vertical line will be the seed moisture content. So as we get higher, it gets the seeds, the moisture content is higher, and as we go down, the seed moisture content is low. So that's the initial moisture content of the seeds. And on the horizontal side, the temperature is higher on the right and the temperature is lower on the left. So we have 4 different quadrants with different kinds of conditions under which seeds are stored. So let us compare. So the first one is the ambient storage, where you can see that the temperature as well as the moisture content is not at all controlled. We have no control over them. And you can see the single dollar sign, which means it is the cheapest, because we are doing nothing. We just have a premise. We are putting the seeds inside them. And then we have the next type, where we are controlling the temperature, but we are not controlling the moisture or the relative humidity. And these are the non-desiccated cold storages. The most cold storages in many places all over the world, seeds are stored under these cold storages. But again, the costs are very high. You can see from the rating there that since we have to go for refrigeration, so the costs are high. And then we have the most expensive seed storage facility, where the temperature as well as the relative humidity are controlled. So they have equipments for cooling, they have equipments for drying the air. So these are used in germplasm storages, in gene banks, and very high-end seed storage facilities. The last one is very interesting. So we don't have any control over the temperature. We are storing it under regular conditions, ambient conditions, but of course not directly in the sun. But we are having a control over the seed moisture content. So this is done under ambient temperature with no refrigeration, or we call it the dry storage or desiccated storage. So you can see the comparison. Yes, the costs are slightly higher than the no control ones, but the effects you'll see that are much higher. So this is called the dry storage or the desiccated storage, or in other words, desiccated hermetic storage. This is the particular storage where what kind of storage which we normally say that it is the most optimal way of storing seeds in a community seed bank. So this is the desiccated hermetic storage. This is economic, effective for medium-term seed storage. We are not going for long-term storage like 20 years, 50 years, or something like that. But for our regular work, storing seeds for 5, 6, 7 years is quite good. So what are the real aspects of this kind of desiccated hermetic storage? So first of all is that we have to dry the seeds. And after drying the seeds, we have to keep them dry. So that's why the word desiccated and hermetic comes. Desiccated means drying of the seeds, and hermetic means sealed or airtight containers. So we have to dry the seeds with some kind of desiccants, as you see here. With some kind of desiccants, we have to dry the seeds, because if you are putting the seeds outside for sun drying, there is a limit till which the seeds can be dried. But if you want to dry them more, you have to use some kind of a desiccant. So we have to use this kind of desiccants. We have to use, after drying the seeds, we have to seal them. We have to seal them so that there is no moisture or air leaks inside these containers. And then we have to monitor the moisture, that is there any leaks within these containers. And then, of course, you cannot use these desiccants for indefinitely. So you have to regen, because the desiccants are also absorbing the moisture from the seeds. And that moisture also has to be thrown out of the system. So you have to regenerate the desiccants, so that you dry the desiccants again under certain conditions, and then again use it to dry more seeds. So ultimately, our objective is to dry the seeds. You have to dry the seeds, make them dry, and keep them dry. And just to give you an example, there are different kinds of desiccants. There are zeolite beads are there, silica gel is there. There are even baked rice is also sometimes used. Sometimes brick dust is also used in Uganda. We have seen in some community seed banks. So anyway, any kind of desiccants can be used, and these kind of desiccants, they absorb around 20 to 30% of the dry weight in water. So 1 kilogram of these desiccants, 1 kilogram of desiccants will take out around 100 grams to 200 grams of water out from the seeds and keep it within itself. So that's how we do the calculations. And just to give you an example, if we have 100 kg of maize seeds and from 14% seed moisture content, so the seed moisture content of the seeds is 14%, and I want to reduce it to 8%. So that means there are 6% of reduction, moisture reduction I have to make. So that means from 100 kg, we have to remove 6 kilogram of water. So that's a simple calculation. That much of water must be removed from the seeds. So from the seeds, it has to go out. go out where? It should be coming out into these desiccants. And the desiccants have a capacity of maximum 20 to 30%, on an average 25%. So we have to use these beads or these kind of silica gel or any kind of desiccant which have a capacity of around 20 to 30%. We have to use 21.5 kilograms. This is a simple calculation which we do, so that to take out 6 kilogram of water, we have to use this amount of desiccants, so that all the required amount of water from the seeds comes into these desiccant. So this is a calculation. So these desiccants, after doing this, after drying the seeds, it can be regenerated, and they can be regenerated in an oven and cooled and then used again many times. So you can see that there are different kinds of desiccants are there. On the left side, even we can use roasted rice. So you just roast the rice on a pan, on a hot pan, and then cool it, and you can use it to dry the seeds. It also dries seeds like anything. And it can be silica gel can be used, and even the zeolite beads can be used. And we have even seen that red brick dust also absorbs moisture to a quite good extent. But this dust has to be very fine. So finer the dust, more surface area, the more water it will absorb. So these are even some, I would say, some scientific as well as some locally usable desiccants that we can use to dry the seeds. And of course, you can use it the way you like. It is flexible. You can either mix the desiccants with the seeds, or you can keep it in a layer. But the best thing that we say is that the picture on the right, that you should use these desiccants in a cloth or a paper bag, which is through which moisture can enter and go, so that it ultimately dries the seed. So you can see on the picture on the right that these are real seeds that are stored in a community seed bank. With, you can see chickpeas there, some sorghum is there, and you can see the white cloth bags which has the desiccants inside it. So you keep the desiccants, just calculate that what amount of drying you need to have. In a thumb rule is around 15 to 20% is of the weight of the seeds of the beads or these desiccants is quite good for drying the seeds within a container. So, but you don't have to keep it inside for as long as possible. For these kind of containers, we say that it is around 1 to 2 days. If you keep the desiccants inside these containers, it is good enough time to dry the seeds. But if there are bigger containers are there, you have to increase the time to around 1 week or 10 days or something like that. I'll be coming to it. I'll be showing you those containers and those things also. But these desiccants are to be used so that it is okay. It is safe enough to dry the seeds within the containers itself. And then the next thing we have to take care is the moisture monitoring. Of course, there are certain containers which have these thermohygrometers already fitted inside them, but you can use these simple PT100 sensors. These are available in the online markets like anything. And even there are some clocks and there are some watches which have these sensors. And these are quite easy, and these are quite reliable, and these are also quite inexpensive things with which you can monitor that if the moisture content within the community seed bank or within the container is going up or not. You can also use moisture indicating strips, and I think some of the community seed banks in this project have also been provided with these dry cards, with which you can monitor the moisture inside the bottles or inside the containers or inside the community seed bank. So in a nutshell, these moisture strips, blue means they are dry and red means they are wet. So you can see from the meters and the color code that is there on the picture on the right, that high moisture content indicates the reddish color, that is the pink, and when it goes down, the moisture content is going down. It is somewhere around it will be the blue color. So this is how the moisture indicating strips are also working. We can use them within the community seed bank. We can use them within the bottles. You can see in that picture where a small strip has been added, and you can see that there are chickpea seeds inside that bottle, and there is a paper packet this paper packet has the desiccants inside them. So it dries up the seeds inside it, and after maybe a couple of days, you can take that envelope out, and you can again regenerate the seeds. And I'll tell you how to regenerate these desiccants. So the scheme of operations, what will be the scheme of operations within a community seed bank? This is very important. We have to take care of this. So first of all, we have to sun dry the seeds properly. The sun drying is very important, provided the outside moisture content is on the lower side. Anything above 80% relative humidity, that is the moisture content of the air. Anything above 75 or 80, you should not go with sun drying. It is better because ultimately the seeds will absorb the moisture. So first of all, we have to go with sun drying. And after the sun drying, we have to use the desiccants for a couple of days to dry the seeds inside these containers. And these containers can be of different sizes. So if you are having smaller quantities of seeds which have to be stored for a little bit longer period of time, or larger quantities of seeds, you can use different kinds of containers. You can see that the white and the blue containers in the middle, they are quite readily available for any kind of lubricants or paint in industry. It is available, and they are also a very good method of storing the seeds in little bit higher quantities. So they come in different capacities of 100 liters, 50 liters, 80 liters, all these kind of capacities. But the ultimate thing is it has to be airtight. So these containers have a kind of mechanism with which it doesn't allow any kind of moisture to come in or go out. And ultimately what happens is that after a few weeks using these zeolite beads or the silica gel or these kind of desiccants, after some time you can remove it from the storage containers, because that moisture has already traveled from the seeds inside these beads. So now we can take them out, and after taking them out, we have to use an oven, any kind of an oven, an electrical oven, or even we have tried in Cambodia and in many places we have tried even wood-fired oven, but that doesn't matter. The temperature we should use somewhere around 200 to 300 degrees centigrade, the normal temperature of an oven. So we can use these ovens to dry these desiccants. To dry these desiccants, we keep them for around 2 hours, and then we cool it, and then we seal it, and there we can use it again. And you can see the cycle here. The red arrows is showing that we can recycle, we can use them as many times as possible. And the next primary thing that is very much important is the moisture monitoring. So we have dried the seeds. What happens if there are some leakages within the containers? The containers which we are storing, the containers are not moisture vapor pervious. What happens if they are also leaking? It is absorbing moisture from the air. So we have to be very careful about that. We either have to use these kind of indicators, or we have to use some kind of a moisture meters. It is not correct to say moisture meters. It is actually a relative humidity meters, or technically they are called these PT100 sensors, but don't worry, you don't have to remember that. So these are very much available, and these are very inexpensive things. You can it uses a very small battery, and it runs for maybe 6 months or 10 months on a small, a very small, these button cells. So these things should be used. The best thing to use are these moisture strips, which are very easy to use, and they are quite accurate, and it gives you an indication of what is the exact situation inside these containers. So moisture monitoring, it is very important. Now, I told you that these beads or these desiccants need reach. So it is similar thing for silica gel, the blue ones that you have seen, and the case is also similar with these zeolite beads. So once these zeolite beads needs to be regenerated. Because after we have used these zeolite beads for drying the seeds, they need to be regenerated. So what we do is we heat the beads to around 250 degrees centigrade for 2 hours. So you can see that in these kind of containers, or trays, any kind of metal trays, we heat the beads for 250 degrees centigrade for 2 hours. And then we cool them briefly, very briefly. Otherwise, in this container, because this metal tray, because this metal tray also stops any kind of moisture entry. So very briefly, because it cools down also very fast, it doesn't need a lot of time to cool down. So after heating, it cools down very fast, and after that we can seal it. You can see that these ladies are putting them using a funnel and sealing them in a airtight container. So those bottles which are there, those bottles are airtight, and you can, after cooling them, you can store these beads, and as and when required, you can store it in bigger containers also, but that container should be airtight. So as and when required, these desiccants are used, put in small packets, put inside the bottles or bigger containers, and then after a certain amount of time, when they have absorbed all the moisture of the seeds, they're taken out of the containers. The containers are sealed again, and they are regenerated using an oven. So this is the whole procedure of how these desiccants are regenerated. Now, showing you the use of of these desiccants to dry bulk seeds. So I was showing small bottles. So not only small bottles, you can see from these pictures, and this is a real community seed bank where the farmers are storing their seeds in airtight drums, these drums, and using mesh bags or cloth bags to store the beads, not to store the beads, but to use the beads inside these containers and then sealing them. And then after a week or so, these bags are taken out and again regenerated. But by that time, the moisture from the seeds has already been taken up by these beads. So what is the result? The result is we are having drier seeds. And now, since we have the drier seed, we have made them dry. We have to now keep them dry. So keeping them dry is the job of the containers. So we move on to our discussion of some locally available airtight containers. And all the pictures that we are showing you right now are real life situations. These are not just some pictures picked up from the internet. So you can see that the picture on the top left corner are very big storage containers which are really airtight, and these they are around 150 kilograms of rice seeds was stored here, and desiccants were there in layers in the middle and on the top, and they had ropes inside them so that they can be pulled out easily. So this was done somewhere in 2014. This picture was taken where you can see that we had been storing some wheat seeds here, not rice, wheat. HW2045 was a variety in India of a wheat variety which was harvested and stored in a seed bank. This was not a well-running community seed bank per se, but farmers were storing using this technology in larger quantities. So there are so many pictures. You can see the plastic containers here. Let me annotate. Something's happening with the annotation, but anyway, it doesn't matter. So okay, actually it's here. This is the plastic container where you have already the moisture meter already fitted. These are the bigger containers. You can even use Coca-Cola or Pepsi bottles. Here also you can store the seeds, and you can see a small pouch of a desiccant lying over here. Here you can see. This is a pouch of silica gel that is being used to dry whatever this is. And you can notice that there is no air gap. So we are also restricting the oxygen. This is not a very good method of storing because you have a lot of air here. But still, if you are using these desiccants, the low moisture content is good enough to reduce any kind of aging of the seeds. So this is some of the locally available airtight containers, the storage containers which we can use to store the seeds. And you can see there are so many different ones. There are ones which are the metal ones are there, which are having moisture relative humidity meters fitted on the sides, on the walls, so that you can see exactly what is what is the moisture content of the, the relative humidity of the seeds inside these containers. So these are the different kinds of containers, and this picture also shows a real life community seed bank where they are storing the seeds under bigger containers as well as the smaller containers. So these are some more examples from India where the seeds are being stored within community seed banks with larger quantities and smaller quantities. And you can see that all these small bottles are having these cloth bags which are having these desiccants inside. So they are here for some time, and maybe after a couple of days they will be taken out, all bulked, and they'll be put inside an oven. And after doing that, they'll be regenerated, then again kept, so that a fresh batch of seeds can be dried. So the main thing is that you do not need to store the seeds and these desiccants together in this community seed bank. You can use it for drying, and then after a certain time you can take them out and make sure that the container is sealed well. So that was a small discussion on the technicalities of seed storage. I will have a few more slides on documentation, but before I go there, do you have any questions? You can please unmute yourself and talk, or you can raise your hands and let me know. Before I move into the documentations, the record keeping and all these things, how to do in a community seed bank. Do any of you have any questions on that? Yes, Taban.
Yes, I have a question on.
Yeah, please.
After using those desiccants, like the other strip, when you wanted to reuse it, like after heating, will those colors, because now when there's some moisture in the seeds, we realize the colors will change from white to blue. Will those colors disappear after heating?
Yeah, okay. So I think there is a small difference in the understanding. Let me go back to this slide. No,
maybe let me give you. I have like 3 questions.
No, actually, those strips are not to be used for drying the seeds. Yes. These strips are only for indicator. You can see these strips.
Yes.
These strips are only for indicating. So these strips automatically, when they absorb moisture, they become pink, and then when they release moisture, they become blue. So these strips also can be used for a long time. Is that your question?
Yes, that is my question.
Okay. Then please.
The second question.
Yeah.
Okay, how will we use the bricks also for reducing the seed moisture content? Can we found it, either we put it like we reduce the shape a bit, then we put inside the container so that can reduce the seed moisture content?
Yeah.
You mentioned we can use bricks, yes.
Yeah. So it is better not to break these beads, if you wanted to ask that question. But these beads come in a smaller size so that even it can fit in a smaller narrow mouthed container also. But what we suggest is, whatever you use, it's better if you can use some kind of a mesh bag or a cloth bag, or even a paper pouch. We have even used pouches made out of newspaper, or envelopes made out of paper, and we have put the beads inside them and stapled it close so that it doesn't spill out, and put that envelope inside the seed bin or the seed box or the seed bottle, anything. But for larger quantities, it is better to use a cloth bag, because otherwise that paper will not be strong enough. The paper envelope may break, but a cloth bag is much better. So it is always advisable that you use any kind of a pervious material like cloth, paper, mesh, even net bags. You use that so that moisture migration can happen. So water from the seeds will come to the air, and again from the air will come to the beads, and it happens. So you should not put those beads in a plastic pouch. because then the purpose will not be served.
Okay.
Okay.
The third question is,
Yeah.
I had this experience. I have some okra seeds I put in this bottles of water, either soda.
Yeah.
Then after some time, I realized the bottle is depleting from the side. It's depleting inside. Then when I open the bottle, also now when air enters, it comes to the normal shape. What could be the reason?
Come on, the seeds are respiring. So it is just like when you are sealing the seeds inside that bottle, there is 21% oxygen, 75% or something nitrogen, and rest gases inside. But the seeds are, when they are respiring, they are absorbing the oxygen, just like us.
Oh.
So when the oxygen is taken out from the air, it creates a vacuum. And the
Yes.
walls of the bottle are not that strong to withstand that, so it collapses a bit. But it is a
Okay.
good sign. So if it is also said, if you, I'll send you some information on these bricks bags or the seed storage bags designed by IRI, the International Rice Research Institute. So there they store the seeds using this kind of technology, where they put the seeds and then they close the bag. It's a plastic bag, but very thick plastic bag. They close it so tightly that there is no space between the seeds, and there is no space, there is no air space. They tightly close it and they tie it with a string, and they keep it like that. And after some time, so these bags have a capacity of 60 to 80 kilograms. So that much of seeds after, and they have a very little space, no space at all, only the space between the seeds, and all the oxygen is, after a while, maybe after a month or so, all the oxygen is consumed. So when oxygen is consumed, I showed you in my first slide that low oxygen also extends the life of the seeds, doesn't it?
Yes.
So, and also since there is less air inside, that means there is less water also inside the air. So everything is taken inside the seeds, and then the respiration of the seeds ultimately goes down. If the respiration of the seeds goes down, the life increases. And also if there are any kind of insects, inside that seed bulb, they also die, because the seeds have already taken up all the oxygen, because they need oxygen to survive too, and the bag is closed in such a tight way that nothing is coming in. So the insects die out of starvation.
Okay.
You get my
Thank
point?
you.
Good. Okay. So
now there's
yes, please go ahead.
okay, like storing these seeds in this envelope. Does it mean that after putting the seeds in the envelope, oxygen cannot enter the same envelope again?
What is the envelope made of?
This khaki envelope. Like when we went to visit during the study tour in the seed bank in Uganda, they have those envelopes that are used for keeping the seeds.
Okay, those gene bank envelopes.
Yes.
You mean the metal foil and the
plastic
one
in the gene bank. Yes.
Those are fully moisture vapor impervious, so you can store the seeds inside those envelopes, provided you seal it, either heat seal it or vacuum seal it.
Okay.
Those are totally, it's as good as a metal container, as good as a can.
Thank you.
Good. Any other questions? Then we go ahead with the documentation. Let me come to the slides. And I really thank Taban for those questions, because these are some very basic thoughts that comes to our mind when we are talking about seed storage and how the seed physiology is affected. These are a little bit technical, but I think we, being professionals, we should know about the technicalities of it also. Anyway, so let us come to the community seed bank documentation. So community seed bank documentation is actually a management procedure. So you remember last time, Abhishkar, in my last session, Abhishkar was talking about the management committee. we should have a group of farmers, a group of individuals who have a common goal and a motivation and an awareness to store seeds and find out that exactly what are the seeds or what are the varieties that are accessibility becomes a big question. We are not getting some varieties, we are not getting some crops also. So that management, the management committee or the management team of the community seed bank takes care of the documentation inside a community seed bank. And it is actually a management of collection that is used by the farmers, and it has accession identification. So accession, we have to get used to this word. So accession means one sample. So whatever seed that is come, it may be a lot. Many of you may call it a lot or an accession, or it may be a small quantity, bigger quantity. So it is called an accession, and an accession consists of a farmer who's to whom that particular variety or the sample belongs, and also the quantity, the variety, the crop, and all those details. And there has to be a proactive management of accession. So the seed bank management team has to be very proactive in the management of this accession, and these accessions should be available for use and exchange. So these are actually the management rules, or the management, the seed bank, community seed bank management should have these qualities so that they can carry ahead with the documentation part. There are actually 4 books or 4 documents that is maintained within a community seed bank. It may sound so many, but it's not. It's very simple. So first is the registration book. Then we have the management book or cards. I'll explain what these are. These are either the management card, we call it. Also, we call it the accession card. Then we have the accession appointment diary. It has big names, but don't worry about it. They're really simple things. And we have a movement book or a duplicate book. I'll again come to it, to the what exactly they are. So we are coming to the first document. So the registration book, or it is called the accession register. We have the same name as it is used in a gene bank. So an accession or the registration book, every sample that comes inside the community seed bank will have an accession number, and it is a unique serial number. Usually the seed bank or the gene bank prefix to differentiate between different seed banks. So if we have a Sanak gene bank, maybe as a community seed bank, so it will start with an SAN. Just giving you an example. And the first sample that enters, the first seed or the first seed lot that enters will be given a SAN 0001. The next one will be given SAN 0002. And then, of course, that number will be unique to that lot. And then, of course, these are the information that needs to be entered, the date of entry, the type of material, it may be seeds, it may be suckers, birds, or any kind of thing, the crop, the variety, and it should have all the names with which it is known. Then the source farmer, maybe the gene bank from whom the accession has come. Then from the locality, latitude and longitude, the GPS location, if we can do the georeferencing, it is very good, from where it has been collected or from where it is coming from. The collector's name, source, known attributes. So this is also very important. So what are the traits that it has? So are the fruits red instead of green? Or maybe it is a short duration variety, or maybe this variety can be used where the soil is affected by salinity. Are these characters are there? Or it is having a very nice aroma for rice? Just giving an example. So all these traits should be there. And some remarks and location of safety duplicate. So suppose we are also keeping whatever the seed was coming, we are keeping it somewhere else also, maybe in the university gene bank. So if it is there, we also entered this kind of information. So you can see that whenever a seed enters, we should have all this information noted inside a community seed bank. And I will give you an example also. So of a registration book where you can see the accession number, crop, variety, the farmer's name or the community, the date of accession. So the date with at what time it has entered the community seed bank and the quantity that has entered. So there will be many others also, but for the time being, this community seed bank had only this information. So this is one example of a registration book. Now we come to the management book. Actually, we call it a management book or management card. So this is called an accession card because this card is always stays with the sample, or it is filed, but it is one card, one sample. This thing, the registration book, it is a book, it's a form of a register where we have all the information in a tabular format. But here we take one accession or one sample should have all this information. So it should have the accession number, so one card or one paper for one accession. So if there are 100 samples, 100 different varieties or 100 different samples within a community seed bank, we should have a file containing 100 sheets of this management book. So actually, it should have an appointment date or the date at which something has to be done on that sample. Okay, and then description, all these things. So better, I will give you an example so that you can understand it better. So you can see here an example of a management card. So this is, let me start annotating so that you can understand this better. So this is the name of the community seed bank. This is the accession number. So this is the same one accession for one card. So this page will only belong to one accession or one sample. And 20th December it entered, and the starting day weight was 500 grams. So it was sent at location A6, maybe on a drying floor of a community seed bank to dry. And then at 1st January, it was to be collected for treatment. And the fungicides with whatever was used. And then again, seeds were left to air dry. Then again, on 1st February, the drying process was done. So all these things, whatever is happening to that sample or whatever is happening to that bulk of seed has to be noted. And also the weight, because you can see after drying, there will be some weight loss. There will be some weight loss. So that you can see that drying process, the desiccant is changed, 100 gram desiccant is used. So everything, whatever happens to that sample is noted. So it gives you a very good understanding of what is. So you are on the top of the things within a community seed bank. You are on the top of the thing, whatever is happening to all the stock that is there inside the community seed bank. And you know, this is a very serious process. We have to take this very seriously. And I would show you again some examples of this community seed banks management card. So you can see that this is the accession number. This is again the another accession number. And this is one card. This is another card. You can see that on this date, it was taken for germination test. This was dried in the sun, sealed in PET bottles. So whatever is there, you have to enter. So you have to enter and you have to write. So that's the work of the community seed bank management committee or the technical person or whoever is taking care of the samples within the community seed bank. Then I'm coming to the third document, and this is the most simple thing. So it is called the accession appointment diary. So it is nothing but it's a desktop planner. It's a calendar. It's a calendar. So whatever information was there in those cards, it is just a person just jots down so that the person knows that every day what are the things that needs to be done. Maybe some desiccants needs to be changed, or somewhere sun drying is happening and you have to take it back inside, and maybe it has to be going for cleaning. Some maybe 4 or 5 different crops has already entered and they needs to be again dried, their moisture checked. All those things has to be written down in a calendar so that we don't miss anything. So it is nothing. It is good to have accession. It is a big word, accession appointment diary, but don't worry about it. It's a calendar. We should have a calendar of operation. We should know what happened, what we are going to do this week or next week or the next day. So this is very important. And again, the seed movement book. So the seed movement book, you are all aware of. It is just a kind of a stock register. So actually it has counterfoil. Suppose it tracks the movement of seeds from the seed bank or the gene bank. It can be a duplicate book to allow the gene bank to maintain a copy. So if it is, suppose from a certain, suppose we have of a certain wheat variety, we have around 50 kilograms, and a farmer has, a member of the community seed bank has taken around 10 kilograms from it. So once that 10 kilogram is taken out of the stock and given to that farmer, seed movement book is signed. It is written that this variety is given to this farmer on this date, and it is signed in, and it is written in duplicate. It can be, you must have seen a carbon copy. So one part of that slip is given to the farmer who has taken, and the other remains. So that is the movement book. So now we have all the information that whatever the seeds have been moved, it may, a sample may go to even to the gene bank for safekeeping. A sample may have also, if the farmer, because normally what happens is that after that, that farmer who has taken that 10 kilograms also returns some to the seed bank, because whatever was taken has also to be returned to the community seed bank. So that movement also has to be noted. So this is actually a stock movement, how the stocks are being moved. So this, with these documents, we keep the management inside the community seed bank very up to date, and we have no problems with stocks. We have no problem with whatever is happening within the seed bank. And next document, we don't call it exactly a document, but you have to be very careful about labeling. So it may be bottles, it may be bags, it may be drums. We have to be very careful about what we are labeling. So it is always advisable to have one label inside the container and the other one duplicate on the top. And within a community seed bank, we have seen that we use ballpoint pens. Many times the color fades. Even permanent markers are also not working many times unless they're very permanent. But it is always advisable to use a 2B pencil. The pencil is the best. If you write things with a pencil, and if you're sure that no water comes in contact or no one erases it with an eraser, you can always use a pencil, because the pencil is made of graphite and it is a very pure form of carbon, and it never fades. It will never fade by sunlight. It will never fade. So this is just some small tips. You should make sure that labels are in duplicate or even maybe in triplicates. It should, one piece should be there inside the seed bags or the bottles, and the other should be outside so that we can see it from the outside. And just before we go into more details, this is a flow chart of operations, and that how the flow of the documentation happens. So the accession register, seed enters the community seed bank, make an entry. So this is all the flow of operations that we should go. So assign an accession number and then create a management card. Then again, update the activity planner. If any seed movement happens, you have to enter it. So this is actually this is how in a flow chart it explains that how we should go about maintaining the documentations inside a community seed bank. And this is again a same kind of a diagram where you can even find out that the documentation as well as the function. So you can see from here that the seed is first registered, it is inspected, then clean, treatment, if sun drying is needed, and then we have to go for testing. So we can take some seeds and do a small germination test in the premises, or even using a small rolled up cloth. We can see that, yeah, if the germination is okay or not. Then we go check the quantity after the germination. If the germination is less than 50%, it has to be sent for multiplication. So these are certain rules within a community seed bank that the management team has to formulate. So, and if it is more than 5% moisture content, we have to go for drying. We have to add the beads. And then after multiplication, after storage, every 3 months, we have to check the indicator papers. If the indicator is pink, we have to go for drying. After 6 months, we have to check germination. And if the germination is is less than 50, again, we have to go for multiplication. And then we have to check quantity. If the quantity is insufficient, again, we have to go for multiplication. We have to increase, and then the stock again is kept inside the community seed bank. So this is actually the flow chart of operations that we have to take care while working in a community seed bank. I will share these presentations and these diagrams, don't worry. And also the recording of this will be there. And so concluding, seed storage must be done under seal dried conditions. So first you have to dry the seeds, and then you have to keep the seeds under sealed conditions so that no moisture comes in from the outside. And every step must be taken so that the seeds do not absorb moisture from the outside. And we have seen that high moisture causes seed death, and also high moisture causes insect damage. And we can use any kind of desiccant to dry the seeds. You can have silica gel, you can have zeolite, you can have brick dust, baked rice, anything. And of course, then we have to go with the documentations. This is one of the community seed bank management tasks. And there are 4 kinds of documents. We have already learned about that. So this is actually the concluding our discussion for today. Before we go into a group work, if you have any questions, please ask your questions, because after that we are going to go into we can go for a 10 minutes break after